Hi everyone, welcome to your chapter 11 video. Much like some of our previous chapters, I actually have decided to divide this chapter into two parts. That's a little bit different than what your syllabus suggests. So my plan is to go ahead and put chapter 11 into two parts. And also that means that if you're watching this in the fall of 2020, then you will have a homework that is due at the end of this week, which I believe will be, let's see, October 1st, 2nd, 3rd. I think that's going to be October 3rd. You have a homework due. And that homework will be exclusively over chapter 11 content, okay? Now let's think a little bit about where we've been so far. So we've gone through 2 units of study and you've had 2 exams. And so far we've thought a lot about cells. We've thought about their structure, right? For example, we've thought a bit about their chemical composition when we studied macromolecules. So let's put chemical comp. And those were our macromolecules. We've also thought a lot about their organelles. So we went through and defined what all the various organelles were in a cell, right? So we've studied a bit about the structure of individual cells. We've also studied something about their function. So we talked a little bit about cell systems. We talked a little bit about chemical reactions via enzymes. These reactions are what allow the cell to function and stay alive. And we've also talked about what's broadly termed energy metabolism. So that's things like fermentation, cellular respiration, and even photosynthesis, okay? So we've talked about structure, we've talked about function, but so far what we've done is we've focused in on individual cells, right? Today, I want us to think a little bit about how cells talk to one another, about how they interact together to perform functions, okay? So let's see here, what else do I want to, okay, so in talking about how these cells interact with one another, here are the things that I want us to focus on. First of all, we're going to need to take a good hard look at the surface of cells because that is what is in contact with other cells. So we are going to focus on the structure of the cell surface a little bit. We've talked about the plasma membrane, but now we're going to talk about features that extend beyond the plasma membrane. We're then going to focus in on how cells adhere to one another, basically how they stick together. And then we're going to talk about how cells communicate, and we're going to break this down into 2 parts. We're going to talk about how cells that live right next door or adjacent to one another communicate. And then we're going to talk about how distant cells communicate. Finally, I'll talk just a brief moment about how unicellular organisms talk to one another. Now, these items here are going to be the focus of part 2 of the chapter, or part 2 lecture, and these items up here will be part 1, okay? I will go ahead and warn you that this chapter is a little bit vocabulary dense, so my suggestion to you is that you avail yourself of the resources that you have. So you definitely have within your Pearson eText, you have some vocabulary flashcards. And you can go in and create your own deck. So it's got these flashcards already loaded, but if there's something in there I didn't talk about, you can actually delete that from your deck and then quiz yourself. So I would highly recommend that you go ahead and start practicing some of this vocabulary. All right, let's start out thinking about the surface of cells, okay? And as you know, cells come in 2 flavors, right? They come in prokaryotic and eukaryotic flavors. So let's think about the cell surface of prokaryotes for just a minute. Now, we've talked fairly extensively about the plasma membrane of cells, and so we've sort of treated that as the external barrier of cells. But the truth of it is, I'm going to kind of draw for us a bacterium here. And so we have a plasma membrane, and we kind of think of that as being the boundary of the cell. But it's actually pretty rare for a cell to be, for the surface to be just a plasma membrane. There's usually some kind of layer secreted around that membrane, and I'm just going to make hash marks. That's not intended to be cilia. I'm just trying to sort of signify for you that there's usually something that's secreted by the cell that's external to the cell that surrounds it. And the reason for that is it assists in protecting the cell. It assists in providing structure for the cell. So let's think about what this external secreted layer looks like in prokaryotes versus eukaryotes. Now recall that prokaryotes, we can divide them into two large groups, right? We have bacteria and we have archaea or archaea. Neither of these have nuclei, neither of these have membrane-bound organelles, but they are structurally different on the interior in terms of like some of their chemical composition. So let's look at their external layers that are outside of their cell membranes. Now, if you are a bacterium, which would be B and C here, then the thing that you need to know is that they typically have a cell wall. We mentioned this before when we talked about cell structures, but let's go back and revisit this. Bacteria have a cell wall, and this cell wall is primarily composed of something called peptidoglycan. Now let's break this word down for just a second. I mentioned peptidoglycan in a carbohydrate lecture, okay? I mentioned it as a polysaccharide that was branched and structural. But this prefix peptido should tell you something about the carbohydrate in this cell wall, and that is that it is associated with proteins. So it's a combination molecule. It's a polymer of both sugars and proteins. And what it does is it provides a nice sort of stiff cell wall. Now, if you are a pre-health student, this is probably going to interest you because this will come up in your microbiology classes. But we often divide bacteria into what we call gram-negative and gram-positive varieties. Where does that name come from? Well, it has to do with the way that we stain bacteria to see them when we look at them under a microscope. There's a procedure that you'll learn in microbiology for staining bacteria using something called the gram staining technique. Gram-positive bacteria can be stained using this technique. They can be colored using the gram stain because what happens is that stain interacts with this cell wall. Gram-negative bacteria are not stained using this gram technique, and here's why. Let's take a look at the structure of gram-positive versus gram-negative bacteria. So let's orient ourselves. Here's our phospholipid bilayer membrane, okay? So this is inside the cell, and this out here is going to be outside the cell, okay? And here we have some membrane proteins that are embedded or anchored on top of this phospholipid bilayer. Here in this gram-positive bacteria, you can see that right outside of this cell membrane, this cell has secreted a layer of peptidoglycan. And then on top of that is a layer of proteins or glycoproteins called the S layer, which just stands for surface. This is the surface layer of proteins. So the big thing to note here is that really the most external feature on a gram-positive bacterium is this peptidoglycan cell wall, as well as some of these glycoproteins here. Okay, glycoproteins are proteins with a little bit of sugar attached to them, okay? Now let's contrast that with gram-negative bacteria. In a gram-negative bacterium, here we have our phospholipid membrane, and we have our transmembrane proteins or our peripheral membrane proteins here. Here is our peptidoglycan cell wall. And what's interesting is that that is sandwiched between another membrane formed by this bacterium. And then on the most outermost layer, you have this surface layer of glycoproteins, okay? So the reason that gram-negative bacteria are unable to be stained using the gram stain technique is that the stain really can't access this cell wall and to stain it. Now, why is that even important? Well, it turns out that gram-negative bacteria, something like 90 to 95 of them, 95% of them, are pathogenic, meaning disease causing, okay? And that's because it's really difficult to raise antibodies against gram-negative bacteria because they, like a lot of their proteins are embedded in this double membrane sort of structure, okay? So a fun fact also, if you're a pre-health major, is that the way antibiotics work is they inhibit often, not all of them, but many of them inhibit the production of this cell wall. And without the cell wall, the bacterium will die. Now let's contrast all of this. So I'm just going to draw a hard line here, if I can. Let's contrast all of this with the situation in archaea. Archaea do not have a peptidoglycan cell wall. So I'm going to put no P cell wall. And whereas bacteria, we find that peptidoglycan cell walls are very widespread. They kind of occur throughout the kingdom of eubacteria. That is not true for archaea. They don't really have any sort of characteristic cell wall that's associated with them. The only thing that we can say about archaea that is pretty consistent is that they do have this S layer, this surface layer of outer glycoproteins, okay? So they still have some features that extend outside of the typical plasma membrane, but there's no single characteristic cell wall. There's a lot of variability in archaea in terms of how their outermost layer looks. Now let's contrast all of this with the situation in eukaryotes. So when we're talking about eukaryotes, of course, we're talking about anything that's nucleated, has membrane-bound organelles, and so that's going to be things like plants, right? It's going to be fungi. It's going to be animals, right? Any sort of algae, anything like that. All these eukaryotes have different molecules that are involved in their external secretions, but they all share a similar structure, okay? So let's write that down. Share a similar construction. How about that? And it looks a little something like this. So basically, the outermost secretions of eukaryotic cells typically are composed of fibers. Sometimes you'll see people say that it's composed of filaments. They mean the same thing. And these fibers or filaments are embedded in some kind of a goo, okay? And that goo has a name. It's called a ground substance. It might be kind of like a gel, or it might be harder. So let's take a look here. This is a great analogy for what the outermost layer on eukaryotic cells looks like. So here you can see that we have steel rebar that was kind of like fibers, and this construction worker is pouring concrete over the top. So this is kind of like the fibers in the outermost region or the outermost layer of eukaryotic cells. And then this concrete is kind of like the ground substance. And each of these fibers or ground substance has a little bit different function, but they do basically protect the cell and they provide structure. For example, these steel rods, the fibers typically resist tension. So pulling and like if you're like pushing and pulling and shearing, this will resist those pulling forces, the tension forces. Whereas the ground substance resists pushing forces, compression forces, like if you were trying to squeeze the cell in on itself. And together, what this does is it protects the interior of the cell and gives it structure. Now let's look at what this outermost layer looks like in plants and then contrast that with what it looks like in animals. So in plants, you probably already guessed that the secreted outermost layer is the cell wall, okay? So down here, you can see that we have the phospholipid bilayer, and then secreted on top of that is the cell wall to provide protection and structure. And this cell wall conforms to the construction that we just talked about. So embedded in the cell wall are fibers made of cellulose. You guys know that plant cell walls are made of cellulose. And if you remember from our carbohydrate lecture, this is a polysaccharide. It is a sugar, highly branched. Okay, we can't digest it. We cannot digest cellulose. As a matter of fact, if you've ever heard the term soluble fiber, when you're eating foods, this is the soluble fiber. Okay, or sorry, yeah, that's right, soluble fiber. So this is the soluble fiber. It passes through your digestive tract. And the reason it's so healthy for you is it kind of acts like a sponge to absorb water and expand and help keep things moving through your digestive tract in a regular way. Now, these fibers are embedded in a ground substance, just like we talked about. This ground substance is another gooey, this is a gooey sugar. It's a gelatinous polysaccharide. So I'm going to put gel. And then I'm going to put poly for polysaccharide. And you may have heard of this one before. This substance is called pectin. I'll write it down here to give you a chance to write it. If you have anyone in your family that makes jellies or jams, they probably have used pectin. Pectin, you can buy like packets of it, and when you're like cooking down your strawberries, you add like some of this extra pectin to the mix, and that's what causes the jams and jellies to gel up and become sort of a semi-solid mixture. Okay, now this, what I just described to you, is actually what we call a primary cell wall in plants. And this primary cell wall is usually formed when the cell is first formed, okay? So it is usually formed during, we'll put during cell development, cell growth. So this is formed like right after cells divide. One of the first things that happens is this primary cell wall is formed. If there's a primary cell wall, then that suggests that there is a secondary cell wall, and that is in fact true. So let's take a look at this diagram. This is obviously some dead plant cells because you can see that the guts are missing, okay? So we've got two of them here. This up here is one cell. This down here is two cells, and I'm going to change my pen color so you guys can see as I draw on here. Okay, so what we have here is that if you look, this outermost region out here, that is the primary cell wall that was initially formed when these plant cells first divided and started growing. Okay, so here's another one where I've got my yellow pen. Okay, now let me change colors again. All right, now let's do pink because that'll show up. All right, so that means that all of this stuff in here up to my yellow line, that forms what is called the secondary cell wall. Okay, and again, the membrane would be in here somewhere. So all of this is stuff that's secreted on top of the cell membrane. So all of this is secreted on top of the cell membrane. All right. Now, the composition of this secondary cell wall, it varies according to the function of the cell. It's still going to conform to the structure where you're going to have fibers embedded in a ground substance. But the type of fibers or the ground substance, that's going to vary. So for example, if you're talking about a leaf, this secondary cell wall is going to have a lot of wax in it, okay? Because remember, we talked about leaves having a waxy outer surface to prevent water from evaporating out through the leaf. If you're talking about a stem, then what you'll find is a whole lot more of those cellulose fibers. Whole lot more of that than even on this outer bit here, because what it's trying to do, these cellulose fibers, what their function is to make things stiff and give the plant support, right? If you're talking about wood, then you're even going to add a second fiber called lignin. And lignin is super stiff, stiffer than cellulose. And of course, that would make sense because wood is super stiff. So plants have a primary cell wall, they have a secondary cell wall. Both of these are secreted on top of the cell membrane to provide structure and support and protection for these cells. There's one other feature that I kind of want to draw your attention to because I'm going to talk about it in a few slides. And that is this region here, this middle lamella region here that I'm kind of highlighting for you in blue. So this right here where I've got the yellow is the cell wall of one cell. Okay, and then here's the cell wall of a second cell. So this middle lamella region, oh, I did that in blue. This middle lamella region doesn't belong to either of those cells. And we're going to see that what this middle lamella region does is it's kind of like the glue that's going to hold these cells together. Okay. All right, so that's what this outer layer that goes beyond the cell membrane looks like in plants. What does it look like in animals? We mentioned this briefly during our discussion of cell features in chapter 7. In animals, this secreted outer layer, so the outer layer that goes beyond the cell membrane, that is called the ECM, which stands for extracellular matrix, okay? It is also going to conform to our structure rules where it's a bunch of fibers that are embedded in a ground substance. What differs in animals is that the fibers tend to be higher in protein and not so much about the carbs, okay? So if you remember, let me go back one. So high protein fibers, I'm gonna put that here. Let me go back to plants. Here, you've got this primary cell wall out here. All of this here is the secondary cell wall, so I'll put primary, I'll put secondary. But the thing that was the fiber here was cellulose primarily. And that is a carb, right? That is a carbohydrate, a sugar. It's a polysaccharide. In animals, the fibers are actually made up primarily of a protein that you've heard of before. It's called collagen. Thought to be one of the most abundant proteins in your body, all right? So collagen are the fibers, and that is in fact a protein. And here's a diagram of it. It's basically just showing you that you have a series of proteins that are twisted together. It's typically about 3 chains. And then you can see that these 3 chains become fibers, all right? These fibers are embedded in a ground substance called proteoglycan. Okay, once again, let's take a look at this word. Glycan is typically a word root that means sugar, while proteo means protein. So you can imagine that this is going to be a molecule that is both protein and sugar. And what you see here in its diagram is a core protein with a whole bunch of sugars tacked onto it, okay? And what happens is if you put a bunch of this together, it forms a gel-like substance, okay? All right, now I have some notes for you about the animal extracellular matrix here, so let's take a look at those if I can. Here we go. So the animal extracellular matrix contains much more protein relative to carbohydrate than does a plant cell wall. Another thing to note is that most of the extracellular matrix protein is secreted via the secretory pathway. So that means that all of the goo that ends up on the outside of an animal cell, so I'm going to draw kind of an animal cell, all of this stuff out here, that is the extracellular matrix. Because most of this is protein, it follows the secretory pathway. That was an exam question for you. And if you remember, the secretory pathway is how we make and secrete proteins. Starts out in the ER, goes to the Golgi apparatus, and then goes to the membrane, where through exocytosis, proteins get spit out. So that's exactly how you end up with all the proteins in this extracellular matrix here, okay? About a quarter to a third of all the protein in your body is collagen. And the last thing I want to note is that the amount of extracellular matrix depends on the type of cell you're talking about. So for example, if we're talking about human beings, bone and cartilage cell, they have very few cells, bone and cartilage tissues have very few cells, but lots of extracellular matrix, whereas the skin has very little extracellular matrix, but lots of cells. And while I've given you a very generalized, this is a very generalized case for what this extracellular matrix looks like in animals, I'm going to write this is in general. It can vary by the type of cell. So for example, you guys know that if you're talking about bone, the extracellular matrix has a high mineral composition, particularly calcium, right? Okay, so that's what the cell surface starts to look like in cells. Now that we kind of know what the outside of the cell looks like, how do we get these cells to stick together, okay? And sticking cells together is called cellular adhesion. So let's go ahead and mention that. Now in plants, it's a pretty easy deal, right? You've got these cell walls. So here is sort of a stereotypical plant diagram, and we've got cell walls, and that's what's being represented here by the tan, the light tan, okay? Those are our cell walls here. Our cell membranes are being represented in the light blue, and then all of this in here is the interior of the cell, right? And I mentioned this before, but this feature that's running in between, let's see, I think I did it in blue before, but now I'll do it in green just to make sure. This feature here that's in between, this is like glue, okay? This is called the middle lamella. Again, think of it as glue. I think your book actually says to think of it as like, if these were slices of bread, then that's the peanut butter in the middle. Whatever works for you, basically it's just a sticky substance. It's actually made up of more of those pectins, okay? The stuff that makes jams and jellies gel-like, okay? So you can imagine that might make a good glue type substance. Now, let's see what I want to say to you. Okay, now we're talking about animal cells and how they stick together. It turns out that it's a little more complex, okay? So they have some features, some actual features that help anchor cells to one another, okay? This is an example of a tissue type called an epithelial tissue. And the reason I mention that is epithelial tissue is made up of cells that are tightly linked to one another because epithelial tissue is tissue that forms barriers. So for example, your skin is an epithelial tissue. Lining your mucous membranes and lining your gut is a bunch of epithelial tissue because what you're doing is you're forming barriers to the external environment. And so what we find in epithelial tissue is that those cells are really, really tightly strung together. They're tightly packed together. So the reason that your book shows you these features in epithelial cells is because it's really important for these cells to be anchored to one another so that they form a barrier to the external environment. So this epithelium here, okay, is actually supposed to be your digestive tract. So this will be the inside of your digestive tract where you have food and whatnot, but you also have bacteria and things like that. So you want to be able to control what gets into the cells and what gets into your body in this region. So now we're going to form a barrier. How are we going to do that? We're going to do that by sticking these cells really tightly to one another. All right, and we're going to use a couple of features to stick these things together. The first one is called a tight junction, okay? And here's what it looks like up close. It kind of looks like your grandma has stitched a quilt for you. So you can think of these tight junction proteins, these are proteins here, as being thread that has been sewn into a quilt to hold two cell membranes together. So can you see that this is one cell membrane here, and this is a second cell membrane here. So here we've got one cell membrane here, and here's a second cell membrane here, and we've sewn them together using these tight junction proteins. Now, what this does is it forms a watertight seal, okay? Because again, we're trying to create, particularly in epithelial tissue, we're trying to create a barrier to the external environment. All right. Now let's look at another type of cellular adhesion, something that's going to stick cells together. And that is this second beast right here, desmosomes. Okay, so again, tight junctions, desmosomes, middle lamella, all these things are vocabulary terms. I would definitely go ahead and start practicing the vocabulary. Now again, here we have one cell membrane from one cell. Here we have a cell membrane from another cell. And hopefully you can see what's happening here is on the inside of one cell, we have a protein. And on the inside of a second cell, we have a protein. But can you see that there's little projections going through here? And what these projections are attached to are some some linking proteins. Okay, sorry. So these are linking proteins. Okay, and these are what's called, let me make this darker. These are what's called anchoring proteins. So essentially what you have here, it's almost like you have got this protein, but you've nailed it to the wall. And maybe you can think of this as like the wood inside the wall, okay? So these things are being held together kind of like with screws and nails to hold these things together. Now, the interesting thing about these anchor proteins and linking proteins is that these anchor proteins are attached often to the cytoskeleton of each cell, okay? So can you see here that the cytoskeleton here is attached to the cytoskeleton here. So if you tug on this tissue in this direction, all these cells are going to follow. And if you tug on this tissue in this direction, all these cells are going to follow because they're linked together, aren't they? Kind of like you've strung them together with a string. All right. So this forms a continuous support structure throughout multiple cells. All right. I like to say that if this is like a quilt, okay, so these are like threads and your grandma's quilted, you can think of these as like rivets in sheet metal or rivets in your blue jeans, even if that's something that helps you. So like really strong anchors. I kind of, like I said, think of it almost as like you've hammered some nails in here into some wood in between to hold these things together. All right. Together, these features basically will anchor these cells to one another. So now what we've done is we've created a situation where in plants, we have stuck some cells together using a middle lamella. In animals, we've stuck some cells together using tight junctions and desmosomes. And once you start sticking cells together, you go from individual cells to tissues, okay? Now, we're not going to talk much about tissues in this course, but that's where this information is headed, all right? But now, we kind of have a little bit of a problem. We have this situation where, for example, here we've glued these guys together. And here we've actually kind of sewn these guys together. But I did tell you that each of these cell types has not only a cell membrane, but some external stuff it's secreted around itself, okay? And especially in plants, you've got some external cell walls here that these have secreted around themselves. So now we have this question of how can adjacent cells, how can adjacent cells talk to one another? Because you can imagine that, for example, if you are a plant, you might want to share, say, nutrients. Remember, plants don't have circulatory systems. So if you are making glucose as a result of photosynthesis and you want to share some with your adjacent cells, how are you going to manage that if you're blocked off by a cell wall? If you are an animal cell, maybe you are, this is epithelial tissue, but what if this was cardiac muscle tissue and you wanted the heart to contract as a unit, not individual cells? You'd want to communicate across these various cells. So we need to figure out how these cells can talk to one another. Well, let's talk about plants first of all, okay? And I'm going to come back to slide 11 in just a second. In plants, what they do is they literally have a series of tunnels that are embedded in their cell walls. These tunnels serve to create links between the cytoplasm of adjacent cells so that they can share resources, they can share nutrients or what have you. And of course, because we name things in biology, these tunnels have a name, okay? They are called plasmodesmata. That is plural. It is plasmodesma if it is singular. Now, be careful. I'm going to go back a slide. Nope, this one. I taught you about desmosomes. Okay, very similar term, not the same thing as this. Plasmodesmata begins with the P because it occurs in plants and it has a different function. It is a tunnel that connects, let's write this down, it is a tunnel and it connects the cytoplasm of adjacent cells. As a matter of fact, you can see here that in most plasmodesmata, you have endoplasmic reticulum extensions that run in between them. so that you're actually linking up not only the cytoplasm, but part of the secretory pathway, okay? So that actually ends up creating a situation where if you were to actually sort of follow a track here, you have a continuous flow, don't you, of cytoplasm that goes all through here and through here. And I bet you there's another, let's see, we'll go out through here. So there's this continuous network of cytoplasm among all these cells because of these tunnels. That continuous network of cytoplasm has a name in plants. It is called the symplast. Okay, sim means together, so that might help you remember that this is the symplast. Now, if you were to trace the cell walls, the external structures, you could also kind of create a tunnel that was where these things are walled off. That is called, the external network is called the apoplast. All right. So that's how we can get next door neighbors to talk to one another in plant cells. How can we get next door neighbors to talk to one another in animal cells? Well, we're going to go back to that original picture that I showed you. So we talked about these two, okay? These two, their function is to stick cells together. This third one that's on this diagram, its function is communication. Just like we saw that there were tunnels that connected plant cells, we have a similar feature in animal cells, only we actually have proteins that are inserted into plasma membranes. So here and here are two different plasma membranes. So this is one cell, this is a second cell, and inserted into these plasma membranes are proteins that link up to form a tunnel, okay? This feature altogether, when you have these proteins linked up like this, is called a gap junction. So for example, if we were talking about, say, cardiac muscle cells and we want them to all contract together, well, it turns out that cardiac muscle cell contractions are due to changes in ion levels. So now we have a tunnel where ions can flow from one cell to another to get the entire heart to contract all at once instead of individual cells. Now, again, be careful. You have a vocabulary term here, tight junction, that sounds very similar to this vocabulary term here, gap junction. So again, my recommendation for you is to very closely look at your vocabulary. And then the last thing that we will talk about is I just kind of wanted to draw a comparison. These are both basically, despite the fact that they have different names, these are just similar strategies for talking to next door neighbor cells. In both cases, plant and animal, what you're doing is you're building tunnels, okay? And you're building tunnels so that these adjacent cells can either communicate with ions. In the case of plants, it might be nutrients. If you're sharing an ER, you might be sharing lipids or proteins. But in both cases, it's the same idea, just a little bit different structure. All right, in chapter 11, part 2, we'll talk about how distant cells communicate. Talk to you soon. Bye-bye.